Amog, you are here? Amog, can you hear me? Amog, you are there? Am I audible, Amog?
You are audible.
Thank you, sir. Amog, can you please speak back? We need your audio to be checked.
Amog is muted.
Good morning, sir. Sir, am I audible?
Yeah, now you are audible now. You are audible now.
Good morning, Manivan sir.
Good morning, sir.
Good morning, Dr. Manivana.
Good morning, sir. Good morning, good morning, sir. How are you, sir?
I am fine. You are busy somewhere for some time, I think.
Yes, sir. All NAC work is going on.
Good morning, Dr. Adil.
Good morning, sir. Good morning, sir.
AVMC, there is some problem with the net connection in between. No, madam is not seen.
Muted news.
Yeah, good morning, Professor Bhatt, Professor
Good
Pushpa.
morning, sir. Good morning.
Good
Shall we
morning,
start,
sir.
Good morning, sir. Good morning, sir.
Good morning.
Sir, good
Our
morning, sir.
speaker.
Good morning.
Good morning. Good morning. Dr. Raghvendra
Speaker
is
is
also
ready.
here, I think.
Speaker is
Sir,
waiting.
welcome, sir. Manivandan, sir. Dr. Narayanan,
Oh,
sir,
nice.
is also here. He
Oh,
is from
excellent.
IIT Madras, Department of Biotechnology, Principal Scientist. From Bio Incubator, IIT Madras. And Adil. Good
Yes,
morning,
sir. Good
Dr.
morning.
Adil.
Good morning, ma'am. Good morning, ma'am.
Good.
Should we start?
Yeah, please go ahead.
Amog. Over to you, Amog.
Yes, ma'am. Good morning, ma'am. So let's start the program. I will start, welcome one and all. I will start the program as stated with the quote by Albert Einstein. The true sign of knowledge is not intelligence, but imagination, which means the motive of life is to think beyond imagination, leading towards innovation. With this quote, I, Amog Deshmukh, second year MBBS student, like to extend a very good and warm welcome to one and all. I welcome one and all for today's program on translation of novel clinical diagnostic technologies. I would especially like to welcome our today's guest speaker, Dr. V. Raghvendra Sai, Associate Professor, Biomedical Engineer, Department of Allied Mechanics, IIT Madras. We welcome you, sir. Before we get into today's program, here is some housekeeping rules for all. Please mute your mics. If you have any concern and queries, post them in your chat box, and all of them which will be answered later. This session is being recorded and will be circulated later for academic purposes. So now I will let's begin with the prayer song for the benediction of Almighty.
What is happening, Amog?
Sir, we are just sharing the praise one, sir. Sorry for the delay, sir.
Okay.
Technical rules.
महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि वसिष्ठवामदेवादिवन्दितमहागणपतिम् महादेवसुतं गुरुगुहनुतं मारकोटि प्रकाशं शान्तं महागव्यनाडगादिप्रियं महागाव्यनाडगादिप्रियं मूषिकवागनमोदकप्रियं महागणपतिं मनसा स्मरामि.
Over to you, Amog. Amog, you are here?
You have to unmute and talk, Amog.
Thank you all. Now I request Ms. Rajarani from second year MBBS to deliver the welcome address. Over to you, Rajrani. Rajrani, please unmute and you can proceed. Over to you.
Rajarani,
Yes, ma'am.
mute
Yes, ma'am.
yourself and talk. Okay,
Yes, ma'am. Earlier I was muted, ma'am. I was not able to unmute, ma'am. Sorry.
everyone was muted by whom? Yeah, now no.
Thank you, Amok. Good morning to one and all present here. As rightly quoted by Steve Jobs, innovation distinguishes between a leader and a follower. On behalf of the management, staff, and institution, Innovation Council, AVMC, it's my privilege to deliver a welcome address. I extended my warm welcome to the bottom of my heart to our guest speaker, Dr. Raghvendra Sai VV, Associate Professor, Biochemical Engineer, Department of Applied Mechanics, IIT Madras. I'm sure he himself, being an innovation, will be a great, enthusiastic, dynamic motivator to all. Thank you. Thank you for accepting our invitation, sir. It's my honor to welcome our beloved Dean Sir, Dr. P.F. Kotur, who is the mentor of a constant pillar of support in the endeavors of ICC. I cordially welcome you, sir. I would also like to welcome our insightful, ever enthusiastic researcher, Director Medical Research, Dr. Vishnu Bhatt, the president of our council. Warm welcome, you, sir. I would like to extend my welcome to our inspirational, dynamic Dean HPE ma'am, Dr. V.N. Mahalakshmi ma'am. I welcome you, ma'am. It's my pleasure to welcome our medical superintendent, Dr. Jai Singh, who's rendered supporting hand at all the needed times. I would like to welcome our DMS1, Dr. Deepa, Dr. Jay Kumar. I'd like to welcome our Vice Principal, Dr. Rajan, who always steers us the right path. I welcome you, sir. It's my honor to welcome Dr. Puspa Kotur, Professor, OBG, and convener of ICC, AVMCH, the person behind the accomplishment of our Innovation Council. I welcome you, ma'am. I would like to welcome our Vice Principal, Student Affairs, Dr. Arunachalam. I welcome you, sir. I also welcome all the esteemed external member of IIC who overwhelming constant support is the driving force of the council. I extend my heartfelt welcome to all our HOIS, HODs, faculty member, postgraduate, and all my dear friends. Thank you all.
We have some our esteemed members of our VMRF. Dr. A. Nagappan is there. Welcome you, sir. And we have one Mr. Jagdish Tunkat. He is actually a person as a management consultant, but he is scientist too. He has done a lot of research in agricultural science. Mr. Jagdish, welcome you.
Thank you, ma'am. Thank you very much. It's a pleasure to be here today.
And Dr. Narayanan, I have already welcomed you. And we have our external members, Dr. K. Manivannan, sir. Welcome you all.
Thank you, ma'am.
Let us start. Let us start.
Yes, ma'am.
Who is introducing?
Yes, ma'am. I request all the participants to switch on your videos. We'll have a screenshot. I request all the participants to switch on your videos, please. Thank you all. Over to you, Amog.
Yes, ma'am. Thank you, Ms. Rajrani, for your hearty welcome address. Now I would request our respected Dr. P.F. Kothur, sir, Dean AVMC and H, to give the opening remark. Over to you, sir.
Sir, your mic is muted. Sorry about it. Yeah. Very good morning to one and all over here. I, as head of the institution, am indeed honored to be present here on this bright morning to welcome today's speaker, Dr. Professor Raghvendra, and all the eminent dignitaries who are present here in the today's kind of webinar. You know, I must acknowledge the presence of Professor K. Manivannan, then the Dr. Nagappan, then Dr. Adil, Dr. Narayanan, who have been instrumental in seeing the seeds, sowing the seeds of this IIC here in AVMC. But for him, this activity would not have been initiated in the past 6-7 months. And looking back at the IIC, I must admire the team led by Dr. Bhatt, Dr. Pushpa, and others, Aarti, and of course the Students' Council students. So there have been no looking back in the past 6-7 months. The IIC has conducted several activities and could get identify itself as a 3-star IIC, and not many medical colleges in the country have ventured into this activity. Certainly we have some advantage of taking a step ahead. And in fact, just about a fortnight back, they had the planning meeting for this forthcoming year. And they have a very elaborate plan for this year, and practically the activity is spreading throughout the year, and they aim to get a 5-star rating. In fact, I hear that they are trying to mentor some of the schools also. That kind of activity has been there. And this platform has been shared by various scientists, entrepreneurs, and the academicians, scholars. And today's one right person, Professor Raghvendra, he's a young entrepreneur and a scientist of, I'll say, international repute. In a very relatively young age, he has got associated with various Indo-UK, Indo-US, Indo-Norway projects, and he has the distinguished. Of course, he will be introduced in detail in a short while now, and he is over here. And for this activity of the students, certainly it will help a long way in the institutional innovation cell. And of course, we have mentoring from the university as well. Mr. Adil, Dr. Adil is here, who is the CEO of the Ganesh Incubation Center. So the wide, so many avenues are over here, and we are very, very happy. And everybody is looking forward for a Saturday now. It will be something kind of a different thing what we'll be doing all the week long. And this kind of an activity certainly has driven away our claustrophobia during this COVID period. And we all feel rejuvenated after these 2 hours of IIC activity. And a couple of thoughts on this, this translation research, or the translation of the diagnostic. Myself being an anesthesiologist, we little acquainted or well-versed with some of these technologies. One thing we must agree that the way the medicine is being practiced has changed because of the research that has happened in the basic research that has happened, and thereby the advances in the diagnostic tools and advances in the therapies, etc. have happened. One thing we must realize here is only the new knowledge or new discovery is not enough to bring in a change in the way the medicine is practiced. So it has to be linked with the insights. What is the insight for a particular drug delivery, particular diagnostic tool? What was the environment? What was the need? So those kind of things have to be linked. Then only the translation will be successful. Otherwise, just doing a discovery. And I am one of the can say humble proponent of evidence-based medicine. I always quote that in the today's patient care, only about 50% of the patients get the best available evidence-based patient care. What about the remaining 50%? They are not getting the best evidence-based patient care. So the thing is, this translation has to occur, taking into consideration the context, taking into consideration the insights. So these are some of the thoughts, those are coming to my mind. And the talks like this will certainly help us to identify the translation gaps, so that the patient care overall can be improved. And we are all COVID, we're very much aware of the molecular diagnostic tests of identifying the viral RNA, DNA, RT-PCR, which has become a kind of a household name for everybody, RT-PCR, this kind of a thing. So this is where we stand. And on this occasion, I am 100% sure Dr. Raghvendra, he is going to share his first-hand experience of, he has a very good experience apart from getting associated with great institutions. He has his own Biosensors Private Limited also. So let his experience help us. And I take this opportunity of requesting him as head of the institution in the time to come. Our team certainly will like to visit his project or the venue, and I hope he will certainly help us. With these few words, thank you very much.
Yeah.
Welcome again. Yeah.
Thank you very much. So, yeah, thank you for the opportunity given to me to speak to you and share whatever that we are doing in the laboratory and what we intend to do in future. So to start with, I know there are a large.
Sorry to interrupt, sir. We'll just have a very good introduction about you, and we shall start, sir. I request Karthik to introduce on our guest speaker. Over to you, Karthik.
Yes, ma'am. Thank you, ma'am. A warm welcome to one and all present here. It's a great privilege to introduce today's our guest speaker, Dr. V.V. Raghvendra Sir. Dr. V.V. Raghvendra Sir, currently Associate Professor, Biomedical Engineering, Department of Applied Mechanics, IIT Madras. V.V. R. Sir received Ph.D. in Biomedical Engineering from Indian Institute of Technology, Bombay 2009 for the work done on development of two novel fiber optic biosensing technologies for detection of biomolecules and pathogens. He spent 2 years of postdoctoral fellowship at University of Idaho, USA, working on development of CS-based DNA biosensor. He came back to India, joined as an associate professor of biomedical engineering, Department of Applied Mechanics at IIT Madras in August 2011, where he established Biosensor Laboratory to focus on development of affordable and indigenous technologies for clinical diagnostic, water, food, and environmental monitoring. Since July 2017, is working as an associate professor. He graduated 4 PhD theses and 2 MS theses under guidance and mentor to SERB NPDF from 2017 and 2021. Currently, 1 postdoc and 8 research scholars are working with him towards PhD degrees. One of them received Student Innovation for Advancement of Research, Exploration, and Gandhian Technology Innovation Award in 2020. He authored 33 pre-reviewed research articles, more than 25 conference presentations, proceeding and filed 1 US and 6 Indian patents. He is also recipient of prestigious Young Engineering Award from Indian National Academy of Engineering and Travel Award from Epson Research Foundation, Japan in 2015. He's been part of several international research grants, including Indo-German Science and Technology Center, Indo-UK, Indo-US, Indo-Norway projects during last 5 years. Based on technological development from his lab, Chem Biosense Private Limited, a startup incubated at IIT Madras Research Park, is spun out in 2018. The IP developed by his team at IIT Madras has been licensed to startups, Recovery Healthcare Incorporation USA and Chem Biosense Private Limited, Chennai. It's our honor to have you, sir. I invite you to deliver your most awaited guest lecture, sir. Thank you, everyone. Over to you, sir.
Thank you very much for the kind introduction. So let me start sharing the screen. So before that, what I would like to do is, I know there is a large crowd here, and then you are all very much motivated medicos, and this makes my job very challenging, mainly because medicos are the smartest people in the world, right? So, all right, but nevertheless, I will try my best to tell you what we are doing in the lab, and then subsequently, so what I would do, let me give a gist of it. We are working on technology development, and I'll talk about the science behind it, and then how we are developing technology. And first of all, before these two, I need to talk about the need, and then subsequently, after talking about technology, what next, what comes next, and then where we are currently. So this is what I'm going to focus today. So I guess now it is 12 o'clock. So how much time do I have roughly?
About
45 minutes, sir.
45 minutes. Okay, great. All right. So,
Hello.
So I'll just play a video to start with, so that you know what we are doing in the lab. This is about 10 minutes. Welcome to the Biosensors Research Laboratory at IIT Madras, where we develop technologies for healthcare, water and food safety, as well as pharma and chemical industries. 20 years ago, we may not have imagined home-based medical healthcare tests. These are blood glucometers, pregnancy test kits, even now COVID detection kits. No doubt we have gone a long way, but we still go to diagnostic laboratories to give a blood sample, and we have to wait to get test results either on the same day in the evening or the next day. But imagine our fellow country people in remote locations such as villages, or you want to have these tests done immediately so that you can actually start the treatment. These waiting times are not really good. So this requires a good technology, and this Biosensors laboratory is actually looking at developing these technologies so that they are affordable, sensitive, they can be deployed in primary healthcare centers. We make use of fiber optic and nanotechnological tools to transform the conventional biological techniques into affordable, sensitive biosensing devices. These sensors work on the principle of simple optical absorption. So the light passing through an optical fiber is absorbed by the nanomaterial whenever the analyte of interest, which is a disease marker maybe, is present in a given sample. We have carried out an extensive research using conventional optical setups, and then subsequently we have gone to compact optical setups. We have realized a proof of concept, and we see that we are able to actually detect analytes of interest down to attomolar concentration, almost 150 molecules in 25 microliters. So this is detected by using a simple pair of an LED and a photodetector. We call this sensor as plasmonic fiber optic absorbance biosensor, in short PFAB. The beauty of this technology is that it is so versatile, it can be applied for a range of biomolecules, including proteins, DNA, RNA, a bacterium, or a virus. Tuberculosis incidence is very high in India. About 30% of cases are never diagnosed due to lack of simpler tests. To address this problem, PFAB technology is exploited to develop a sensitive TB screening test from a urine sample. As we all know, a reliable COVID diagnosis requires RT-PCR, but it is labor intensive and time consuming. Using PFAB technology, we have developed a low-cost, rapid, and sensitive COVID antigen test. Preeclampsia can be treated to save the life of mother and child when diagnosed early before 20th week of pregnancy. PFAB-based screening test for early diagnosis of preeclampsia is under development. Vector-borne diseases such as chikungunya and dengue can be life-threatening. PFAB technology is highly promising for sensitive, accurate diagnosis for a timely treatment. This versatile sensor platform is highly suitable for the development of point-of-care diagnostic devices for a wide range of diseases at primary healthcare center level. Food and water contamination is an important concern for regulatory authorities as well as food processing units. The standard cell culture techniques take a long time to give out results. To overcome these limitations, we have come up with array fab technology for detection of multiple pathogens at a time. Detection of toxic heavy metal ion contamination requires sophisticated mass spectroscopic techniques. A fab-based technology is developed as an alternative with the help of a nanomaterial selective to these heavy metals such as lead, chromium, copper, etc. The fiber optic refractometers developed in the lab can be used to measure fat content in milk. They have potential to help the farmers earn a right price for their produce. Water contamination with endotoxins is a big concern for pharma industries and research laboratories. We have developed a strategy to detect this contamination using our PFAB technology.
The sensor probes are the workhorses of technology. We make these probes using simple optical fiber. We bend them using a machine we developed in the lab. We call it as fiber bending machine. We have developed a mechanism to hold these fibers in a cartridge so that the user can actually handle them in a very convenient way. Subsequently, these probes are subjected to chemical processes so that these bioreceptors for disease markers or pathogens or any other application can be modified. Chemical processes for large-scale modification, surface modification of these probes is already in place. And subsequently, we have developed point-of-care devices, very handheld, compact devices. And at the same time, we want to detect multiple analytes simultaneously in a given sample, or multiple samples, detect them parallelly. So for this, we have developed an array fiber optic absorbance biosensor. We call it as RFAB, which is a compact device that can actually have multiplexed analysis about 8 samples or 8 analytes at a time. RFAB also uses a simple light source and multiple detectors, photodetectors. And we look forward to use this RFAB basically towards ovarian cancer detection or vector-borne diseases, and especially waterborne pathogen detection or foodborne pathogen detection. Fiber optic refractometers are simple devices that enable measurement of refractive index of a given liquid in real time. These refractometers utilize very small volume, less than 25 microliters. They can be utilized in a process industry for real-time monitoring of processes, or even for fruit juice monitoring, milk quality monitoring, or many other similar applications. To translate the technologies developed in the laboratory to bring them to the market, we have actually spun off a company by name Chem Biosense Private Limited. Chem Biosense is an R&D company mainly focusing on these technologies, making them viable for scale up, as well as identifying the most suitable applications for immediate use.
All right. So it's a long video, but it gives an overview of what we have been doing. So I hope you are able to get something out of it. So as we look at this technology, there's a long way to go, and then and also there is an important point I want to make is that there are many technologies that many times they may not really see the light because some or the other problem will be there with some technologies, and that's how it is. So we are yet to figure out how these things will be, whether they make a hit or or basically may not be so much useful. All right. So coming to this science behind this one. So it's important that we actually look at So it's important that we look at the science and the way the technology has been developed. It serves you as an example because any path that you want to take as far as technology development is concerned, it all starts with a problem and then a good scientific phenomena and then a good technology. So what we have done, what we want to do actually is to see if we can convert some of these lab tests into a home care based one or a primary health care center based tests. So that's the aim. And why we want to do it is basically the lab tests that we have are not really within the reach of every Indian. Okay, so for that reason we want to develop. You call it as point of care technology or affordable technology, or a technology that requires a minimum skill is what we want to look at. Of course, you all are aware of it, and pregnancy test kit is something that is already there in the market. Now COVID has come, COVID kits have come. Of course, also if you go to emergency, there are many applications of this lateral flow assay technology as far as a diagnosis is concerned, yes or no diagnosis. Unfortunately, the way it is so simple, it also has its own limitations with respect to, for example, low sensitivity analyte detection is somewhat, the sensitivity is low, the dynamic range is also limited, and you sometimes if you want to do quantification of analyte where it is very important to distinguish whether the disease condition is typically described as an analyte concentration more than this or an analyte concentration less than this, like that. So it's quite difficult to really go for a quantification of these readout, these lateral flow assay devices. So we wanted to really look at some technologies that will be able to, help us in doing things better. So we looked at, there are many sensor technologies, but we thought we want to look at optical sensors, mainly because we have been using optical sensors, optical sensing technologies in the diagnostic industry already in some way or the other. For example, ELISA or CLIA, these are typically absorbance and luminescence based diagnostic technologies respectively. So we wanted to look at, but if you notice, you may not be able to have an ELISA plate for detection of multiple analytes, unfortunately, because there is always a worry of cross contamination when you try to incubate wells with different antibodies. So that's where you in fact you see that there is a need for very good sensing technologies for single analyte and also for multiple analytes or multiple samples at a time. So we thought this, why not this optical fibers, mainly because these optical fibers can be they're made up of silica, and you can draw this kilometers of length without much issues. It's very much established. So we thought we'll work with optical fibers. What is the beauty of these optical fibers? So whenever there is a total internal reflection that happens, there is a small light appears at the interface. And what happens is this interface, at this interface, if you have this fiber, optical fiber has got two parts, at least three parts, in fact, core, and then core covered by, core is where the light propagates from one end to the other end. And this core waveguide or cylindrical structure is covered with a cladding that actually protects the core as well as allows the light propagation without much difficulty. And then there is a buffer layer on top of this cladding layer. It will offer strength to the optical fiber because silica, they are made up of silica. The silica fibers are quite fragile. So you need a polymeric buffer. So, but if you want to access this evanescent field, you need to declad the fiber. That means you have to expose the core, fiber core. When you expose the fiber core, and if you can actually put a fluorophore or like FITC or any chromophore, such as a dye, what happens is the dye is going to absorb the light. Okay, the light passing through the fiber. Mainly because the light is kind of available at the interface and then goes back into the waveguide wherever there is a total internal reflection. Okay, so that can be exploited in many ways in sensing applications. But one issue is that this straight optical fiber has some evanescent field, but it is probably not as sensitive as you probably need it. So if you can modify this optical fiber, geometrically modify it, and that way you will be able to improve the evanescent power. So that means the light available at the core. Okay. So what we have done is to actually make these optical fibers into a U-shaped fibers. Okay. So there is a little bit of chemistry here. Silica, the silica surface, glass or silica surface is functionalized with amines and then dipped into gold nanoparticle solution while observing the spectral changes with the help of an LED and a photodetector, spectrometer. So one beauty of this sensors is that this U-bend fiber is that as the nanoparticles go and bind to the fiber surface, you actually get a nice spectral changes. I mean, increase in the optical absorbance spectrum as more and more particles bind to the fiber surface. So that means it is working as a very good sensor, mainly because these gold nanoparticles have optical absorption properties, and these optical absorption properties of the gold nanoparticles are faithfully reproduced by the optical fiber here. More number of nanoparticles bind, you see more absorbance detected by the fiber. So that's well and good. So why this is really happening is because assume that you are going on a highway and you are basically going at a speed of 100 kmph, kilometers per hour, and there is a sharp turn. Okay, so when you try to take that sharp turn, what you expect yourself is to basically just almost hit the outer surface and then go. So you are coming entering here, and then you basically have to all of a sudden take a turn, and you basically are almost at the outer interface. So in the same way, the light that is entering into the bend region propagates and hits some light does hit the outer curvature and then goes out, but a significant amount of light, that is 60% of light, continues to propagate further into the optical fiber. So this available light at the interface is the reason for such high sensitivity. All right. So this is the science behind these probes. So what is the technology behind the probes? First of all, okay, I'm still not done with the science behind the probes. One beauty is that maybe a few tens of nanoparticles, if they come and bind, you may actually see a very good change. These nanoparticles, gold nanoparticles, have a good optical absorption properties. That's the reason why they are used in lateral flow assays. So when you put the same gold nanoparticles on the fiber surface, you see a good amount of optical absorption signal, even for a few nanoparticles. All right. So then what we did is basically we performed whatever sandwich assay that happens in a lateral flow assay on a U-bend fiber. Okay. So just on a human fiber, we have tried to form these the sandwich immunocomplexes. What we did is the following. We took a sample, a model analyte, added gold nanoparticles coated with antibodies against this model analyte. And we also have immobilized another set of antibodies that are specific to another epitope of this model analyte, so that the sandwich immunocomplex can be formed. So the procedure is simple. Take sample, add the reagent, keep it for 5 to 10 minutes, and then introduce the probe, this U-bend probe, into this mixture. Okay, this U-bend probe is actually coated with antibodies, and then leave it for maybe 10-15 minutes and start observing the response in real time while the sandwich immunocomplex is formed. So this is the optical setup, very simple. This is the optical fiber. All right, this is the solution. So these are bare fiber adapters, these two. So just to handle bare fibers, we have used these clamps. And then the LED that we have used is more or less a normal LED, a green LED. And then this is the photo detector. There's no other big equipment behind the screens. Whatever is there, it is just a small handheld data acquisition card on the photo detector side and a laptop. On the green LED side, there is a LED driver circuit, a small one, that is powered by a mobile charger. Okay, so that's a simple setup. We just want to keep it simple, mainly because, in order for you to develop any technology, it has to be as simple as possible. So to our surprise, we see that when we took human IgG as analyte, and we just took 25 microliters, and the concentrations tested were 1 femtogram per ml to 10 power 6 femtogram per ml. That correspond to almost 6 to 10 attomolar. The 1 femtogram per ml of human IgG correspond to about 10 attomolar, 6 to 10 attomolar concentration. So the results indicate that the 1 femtogram per ml for analytes of concentration 1 femtogram per ml, they give a distinguished response in comparison to what the control is. The control means basically no analyte, no human IgG. So we see that this 1 femtogram per ml of human IgG concentration is able to elicit a distinguishable response. So very nice result, mainly because if you recall what is 1 femtogram per ml or 10 or 6 attomolar, what it corresponds to is about 150 molecules in 25 microliters. Okay, so that's very important to know. So just to confirm whether that's indeed right or wrong, so we utilized this gold nanoparticles. They have catalytic property. This catalytic property of gold nanoparticles can reduce silver around them. Okay, we did the same, and in doing so, why we did so is basically we get about 25 times enhancement in the signal. Okay, so that's the reason we did do that enhancement, and we indeed see that the concentrations are really distinguishable as far as the response is concerned. And these results are verified with standard experimental setups, which cost about 1 lakh to 1 crore to 2 crores. I'm talking about something Biocore SPR, biomolecular interaction analysis applications for these biomolecular interaction analysis applications. We did that equipment, and you see under the same conditions, we tried to see what is the response given by this equipment. We do see that 1 femtogram per ml is indeed that concentration, and it is able to pick up. So another beauty is that by changing the gold nanoparticle size from 40 to 13, you can actually steer the dynamic range to higher concentrations. That's an advantage. All right. So just a comparison with the famous biobarcode assay that was published in Science 2003, of course, almost 20 years ago. But why are we talking about this study? This study was the first one to actually demonstrate automolar analyte detection. All right. So if you look at this technology, it uses multiple amplification steps. There are at least 5 steps, right? And then a lot of reagents are also involved. Instead, if you can actually think about, if you can actually have a very good technology, you will be able to bypass all these processes by simply using a good combination of the nanoparticles and a sensitive transducer in the form of a U-bent probe. A simple incubation and a 2-step incubation process without any need for even washing. That is something very, promising. Why you don't need washing? This basically means these optical fibers, the Evanescent field is a near field phenomena. That means anything in the bulk, in the bulk solution, there could be gold nanoparticles, but this bulk this bulk solution doesn't have any effect on sensor reading, thanks to the Evanescent field, which is hardly 1 micron in the. It has depth of penetration of 1/10 of the wavelength, or at max, it has a field of influence max 500 nanometers. All right, this is the technology, and what about user friendliness? So WHO, World Health Organization, has given a criteria for diagnostic devices for especially HIV testing. This is meant this was prepared keeping in mind the African countries where you really sometimes don't even have electricity. So they say that the devices that one can develop, one wish to develop, has to be affordable, sensitive, specific, user-friendly, rapid, equipment-free, and deliverable. Okay. So yeah, if you ask, this technology somewhat comes close to, this proposed, not proposed, but suggestion made by WHO. So we went for TB. We used MANLAM in urine as a TB marker, biomarker, and we do see that the sensors are able to detect in buffer conditions from 1 femtogram to even 100 picogram per ml. The actual patient concentrations in the patient samples is typically could be urine samples could be between 1 femtogram to even 10 picogram per ml. All right. So we did some more work on the technology. We also did development of gold nanoparticle based sensors for different applications, label-free applications. You don't need to, as of now, you have 2 parts. One, you have to have a probe and you have to have a reagent. So the another strategy as far as this diagnostics is concerned, people have been asking this question whether we can have a label-free sensor. For example, in ELISA, you have enzyme label. In CLIA, you have the same enzymes, but some substrates that gives rise to luminescence. Can we actually have an assay that is free of this reagent? Well, this is called as label-free assays. Even though some people differ with the idea of having a label-free assay, the label-free assay could be very important for certain applications where you are not really specific. You are not particular about concentrations of the analyte, but you are just looking at presence of this analyte in a given sample. So several of these studies are done. We make use of core shell. We prepared core shell nanoparticles for CRS, and then we tried characterizing them. We did discover a lot of interesting phenomena here, but we are still preparing the manuscript because there are a lot of unanswered questions. Yeah. So coming to the technology, the U-bend probes are nothing new. They have been existing almost 1990s. But what is it that we did that is different from what others have done? We tried to develop a technology that can, in fact, make these U-bend probes large in number, and also to be able to use these U-bend probes for diagnostic applications. All right. So conventionally, we have been using a candle and to bend these probes. These fibers, optical fiber core is made up of 200 microns. Okay, so it's a very small fiber, and you can actually use a candle or anything else, a flame or even a laser, to bend it. So what we have done is we have developed in-house a fiber bending machine, we call it as you have seen in the video. So this actually helps in scaling up. Most of the work done in India as well as elsewhere is limited by an important factor, that making these probes is tough. Okay, earlier, if you want to handle this 200 micron fiber and you want to bend it, you really have to practice for about half a day to be able to bend it manually. So, and then you do have inconsistencies as you go for manual bending. So we went for an automated process and tried to improve the repeatability of these probes. Now we are able to get up to 80% of the probes are good. So the reliability of this machine is up to 80%. So, and then so these are we have started using several other applications, several other strategies, such as using a butane flame, which is readily available in a lab. But we thought we see that this flame-based ones are really kind of dangerous, and instead we went for CO2 laser-based irradiation and melting. Okay, this is the point of care device that we are talking about. So here you don't see any display, but mainly because we are doing research and we are validating it. So we thought we should be able to validate it first for some use cases, and then probably would like to go for all bells and whistles such as display, alarm, and all, right? So yeah, this is the RFAB. The design of these two is somewhat very much different. I would say mainly because you have 8 probes to be illuminated at a time. 8 probe sensitivity from intensity from 8 probes has to be measured by the photo detectors. It's a tedious process. So we have spent about 2.5 years with not much of success, and then subsequently we figured out how one can actually make these probes better, and then use these probes using a fiber optic array sensor. So this array sensor really took us about 3 years and still under development, mainly because we want to make sure that this is reliable and affordable. So what all can you do with this is one can actually do label-free sensing, labeled plasmonic sensing, as well as event absorbance sensing. So yeah, we hope that we'll be able to have. Now you see 8. We want to see whether we can actually go for 96 probes in parallel. So that is quite gigantic mammoth task in front of us. But there have been some labs where they have used, for example, seahorse. Seahorse is now part of GE. And they actually have come up with a 96 well plate system where you will be able to measure oxygen concentration from each well, okay, separately. So, all right. So these are, we want to go there mainly because these probes do offer very high sensitivity, unlike many other probes that we are aware of. And then, a large number of applications are possible: label-free, label plasmonic sensing, event-centered web-based sensing. Large possibilities are there. So, applications-wise, the technology, so sensor design is the first part before you think about application, and then the device deployment. So, sample. Basically simple instrumentation, prototype, and other things are all there. But most important is if you cannot make it affordable and field deployable, nobody is going to look at this technology. So that's where we are fine-tuning the technology so that this can be made more affordable. So this is the group. They have been working hard. I must thank all of them. Acknowledgements to all my group, as well as the collaborators and the funding agencies. Thank you very much. So now, I know this has been very technical talk, but how that this is showing an example of how the technology is being developed. Once the technology is developed, one major problem is you need to look at commercialization. So that's again a different ball game altogether, mainly because you really need to jump into a territory that you are not very much aware of. So we did incubate a company, Biosensors Private Limited, in 2018 October, and then 2019 was okay. We are trying to gear up, and then 2020 Corona came. So as many of the people have got affected, the progress of the company also got affected. So we were supposed to go for tuberculosis diagnosis. That's the first bet that we were doing, but we couldn't go as such. So the idea is to develop this tuberculosis test and then take that success and take it for some other diseases such as preeclampsia, and also some other applications such as environmental monitoring, like waterborne pathogen or foodborne pathogen detection, and then pharma applications such as biomolecular interaction applications. So that has not happened so far, but hopefully we will be doing it as we go along. So you need to form a big team. If not big, it's a small team, but capable people. So our technical in charge, our engineer Swami, he is the one who made all the machines in the lab. Any machine that you see in the video is made by him. So a very important contribution to the lab. And he is Divakar. He has basically developed TB assay, COVID assay, right? So we in fact have a partnership with the US startup where they were supposed to take up these COVID tests if he was successful. Unfortunately, we didn't get a chance to go for clinical samples thanks to the either we don't know how to operate it, or we were not really able to connect with right people who can give us samples in right time. And the probes that you see are typically made by our technician Satish. So this is a good team. Once you have the team in place, you can imagine, you can think of starting a startup, right? So these are really the people who are making it, making things possible. So, and then when you want to develop a technology and translate it, you have to choose an important problem that has promise. That's quite promising. And TB in our country is a very challenging disease, mainly because we have 25% of the total global burden in India. And for Indian government also, TB is very critical, disease that government wants to control, mainly because, MDR and XDR TB is on rise. So what are the difficulties? You should understand first whether whatever that you are pitching, whether it makes sense. Yeah, here in this case, of course, it may make sense because the cell culture tests or NAT, they are either too time consuming or a bit expensive. And also you have to see, you may not be able to compete with the existing techniques, but you may have to create a space for yourself, for your technology. Here in this case, we are proposing a screening device for TB. Okay, we are not trying to compete with the existing technologies, but we are proposing a screening test. People coming up with some cough and some symptoms that indicate TB, they can actually undergo this simple test, and then it will be a fast response, and then it will be very clear. If the test comes positive, then they may have to go for an additional test, right? So that's how we were proposing. So, and then subsequently, the background is something that's very important for the technology. Before you can think about commercialization, everybody will ask whether this is scientifically validated or not, whether the technology is scientifically validated. Yes, this at least there is a proof of concept in the laboratory level, and now we have to get a proof of concept at the clinical level, clinical samples, clinical testing. And then subsequently, we have to go for pilot scale, where a large number of samples need to be handled. And there is definitely, you need a lot of money. And unfortunately, there are not many funding agencies that actually support such kind of clinical trials, unfortunately. So if a startup has to rely on some public funding. And fortunately, we are recipient of one of the recipients of TANSEED grant, about 10 lakhs over 2 years. About 19 startups have been selected in the last year, and Honorable Chief Minister has given away the checks to the selected startups just before Christmas. So we are starting, we are utilizing that money to basically look at these devices, make these devices. We do have some devices at some level. We want to make it rugged. So once we establish this pipeline, and then we are ready for clinical trials. Otherwise, we are not yet there. So all these technologies, people have to be in place. Technology has to be in place, funding has to be in place, and then you go for clinical validation. So that's where we are going. We are looking at some partners who can actually partner us with clinical sample handling. Very difficult choice, and of course, more importantly, IP. you have to make sure that you have the IP license, you have license to use. So these are also there. And okay, TB may be lasting for maybe 10 years max. After that, if the test will not, if this technology doesn't work for any other things, that means it's a downfall, and nobody is interested in a technology that will only serve one single purpose. So we should be able to gearing up for many other disease diagnostic applications as well. So, and maybe we want to compete with the existing technologies that time. Yeah. So with that, I stop here. My time is also up. I took 3 minutes extra, but this is mainly to make sure that I complete the story. What is there? Science is very interesting. Technology is even more interesting, but painful. The clinical trials are the most painful things. And then I don't need to talk about the business here, which even I am also not aware of. Yeah, thank you very much for your patience. Yeah, if you have any questions, please feel free to ask.
Any questions, any comments?
Can I ask something?
Yes, sir.
It's a very interesting talk. I think you have done a lot of work in a young age. I think it should encourage our students. Now you have developed several diagnostic kits.
Not yet. We have a laboratory proof of concept.
Okay, so you are talking about like a glucometer and other things, the remote.
Yeah, we want to go there. We want to go there where we can actually make devices that are similar to glucometers. We are starting with TB. We want to do the clinical sample analysis. As I said, we have spent 10 years in technology development.
My question to you is, see, there are multiple conditions which you are doing. So we may have to develop separate equipment for diagnosing each condition. Suppose in a remote area, if you catch these signals from the specimen, whether the diagnosis can be done at a remote area or that is possible. Suppose transfer of information. So you take the signal from the periphery, but diagnosis may be made at another place because the machine may be costly.
Right, that is already there. The current diagnostic model that we have is you have the blood sample, you just in a dry form, it is transferred to a laboratory, and then the laboratory somewhere 100 kilometers away or even 1,000 kilometers away, the test will be done, and maybe 1 or 2 days you will get the tests. That's a possibility. But one of the major issues that we have is if the time is important, and if you want to really make the healthcare delivery fast enough, so that's when all these technologies come into picture.
Yeah, this technique like filter paper is already existing. I was thinking whether the signal can be picked up in the periphery and it can be transmitted to a machine like your IIT or somewhere. Then immediately you give a diagnosis, just like a glucometer. So the glucometer equipment is there, and he puts the blood drop and gets the result. Suppose you want to do multiple tests in a blood, your sample is put, the sensor is put there in the primary health center or wherever it is, and that is transmitted electronically to your IIT or any place where you make diagnosis. That can be done.
Can be done. In fact, it's data transmission is very much easy. What is more difficult is performing the assay.
Another thing about the sensitivity and specificity of these tests. Now, this COVID antigen test we are doing. So many people are positive with the antigen test, rapid test. Then when they go for RT-PCR, they say it's negative. Or other way around, they say negative, then they become positive. So this sensitivity specificity also may be important. Whereas the screening test may be fine. But AD screening test should have high sensitivity, maybe little less specificity does not matter. Should pick up as many diseases as possible.
Yes.
So this may be one of the issues in these rapid tests which we are doing.
Right. Right. We are developing an alternate technology platform that can be used for several diseases, not just one. So yeah, we hope that this technology will be, once it is successful, it will be picked up by the diagnostic industries within India and abroad.
Thank you.
What I wanted to ask is, are there already probes existing, that PFAB, for preeclampsia, for chikungunya and all, or you are still in a developing phase?
We are in development stage. So we have funding for preeclampsia.
And later on, you want to use it in a clinical setup as a clinical trial.
Right.
That's what I was thinking. That it's really very amazing to think like that. That at point of care we can diagnose certain things which we have to wait for.
Right. Yeah. So if we can do it, that will be great. In fact, we did a survey of gynecologists, surveyed with gynecologists, and they seem to say that, okay, probably if they have a test, they will encourage a test for preeclampsia for every pregnant woman.
Being gynecologist, I'm really interested in that, because till today we are still getting those eclampsia tests. So if we diagnose preeclampsia, because there is no such screening test available. So this test can be like screening test we can use if we can use it at periphery. So that is one thing. And another thing, malignancies, we always get to patient at end stage. So that is really wonderful if we can diagnose it earlier. So that's what I was thinking, looking at all these things. It's really, we are hoping to see a better future for our patients.
Yeah, hopefully.
Any other comments, questions? Anyone else want to ask anything? Amok, over to you.
Currently, there are no questions in chat box. So
Okay.
So I would like to thank our speaker for their insightful words, and also starting with the wonderful explanatory video and enlightening us on PFAB technology. Now, I would request Dr. Pushpa Kothur Madam, Professor of Obstetrics and Gynecology, Convener of IIC and AVMCH, to continue this event with her briefing. Over to you, ma'am.
Okay, thank you.
Can I say something? Yeah,
please.
I'm the Professor Raghvendra. I have my words of great appreciation for you for so many things. Even though we are in the medical field, the recent developments which you are being yourself a biomedical engineer, you have certainly ignited the minds of our students. I can say that, these are all age-old common things, tuberculosis, where we are these newer diagnostic, even myself being so many senior teachers, and we are not updated ourselves. So thank you very much. It was an excellent presentation and information sharing, knowledge sharing by you.
Thank you. Thank you very much.
So I have already expressed that it's really amazing to know that our patients will be benefited so much by these technologies. And Dr. Raghvendra really took us for virtual tour by showing that video, how do they work. That is very interesting thing, what I felt. And with this, using these technologies like fiber optic biosensors, where they use nanotechnology, gold, silver, different particles, to improve the diagnostic capacity of that particular test. More sensitive they will make it. And more than anything, those small probes can be used for small sample size. That is very important. Especially in tuberculosis, we used to, as a postgraduate, as a student, as a junior doctors, we used to keep one bottle by the side of patients, this thing, and we used to tell that 24 hours sputum you have to collect, and sometimes that was also not very helpful. So we are just doing that, and that too, tuberculosis has come back recently because of HIV's upsurge. Tuberculosis, incidence of tuberculosis is also increasing. So you are just diagnosing it in the urine. That is really very interesting.
That is something that we want to do, madam. Yeah.
Yes.
We are yet to test the clinical samples.
We will do that. And I'm sure that it will be definitely helpful for our clinical clinicians, like. And not only for only these diseases we are using it, you are using it for even diagnosing contaminants and vector-borne diseases. And so even we can have a safe water or safe food. About that also we can assure to our people. And more than anything, it is affordable, and you are taking this technology to the periphery, where people don't have any access for tests. Our 70% to 80% people, they stay in rural area, they depend upon PHCs for care. And if such point of care devices are available at PHC level, it will definitely help us to screen certain diseases like tuberculosis, preeclampsia, maybe early stage of malignancies. It's a wonderful thing actually. And how we are trying to translate it commercially, that also little bit you have explained, but maybe you are at such stage that you are not only in startup stage and not at in marketing stage. Yes. And that you can do only after validation and conducting clinical trial. I think our AVMC should be one of the partners for you, so we can collaborate and we can do such clinical trials. And definitely our Dean sir, our Director Research, Dr. Vishnu Bhatt sir, will be a supporting force for this. Sure, madam. I'll be looking. Yeah. So thank you very much, Dr. Raghvendra Sai, for your wonderful that feel of knowing everything about this lab technologies. We are slowly getting because of that video, your talk, and you explained very beautifully, very nicely explained each step. So thank you very much. And another important thing is that is affordable, sensitive, specific, user-friendly, rapid, equipment-free. All these criteria of WHO's this thing you have applied and you are using it. So thank you so much. And another one more thing, how you have made affordable also, you have told that in-house you have designed some fiber optic vending machines, then your own applications you have used. All that really brings down the cost of the test itself. And that is very important for we Indians. So thank you so much. So thank you, Amok, for giving me opportunity to summarize the whole talk.
Yes, ma'am. Thank you, madam, for summarizing. Now I invite Dr. Vishnu Bhatt, sir, Director Medical Research and AVMCH and President of IIC for closing remarks.
At the outset, I have to congratulate Dr. Raghvendra Sai for a very illustrating and illuminating talk. And I'm sure that most of our faculty members and students would have been stimulated by his talk. And once the COVID is better, then probably we'll visit your laboratory, which will stimulate further. See, nothing like seeing in the real life than more than seeing in the pictures. But definitely it is impressive, and I am sure that at least some of our students will take the lead and think of some issues which they can work. And I am sure that you will support them whatever help they need. And as Madam has said, we should develop some long-term plan between the engineering and medical group. Of course, in one of the important thing in our university, there are so many medical colleges and engineering colleges. Only thing is they are scattered in different parts, so that sometimes the communication becomes a problem. I think with the COVID phase goes off, then probably interaction may be better. I'm sure that our students will be benefited, and with your talk and enlightening them, something will come out which will be useful to the future. So thank you very much. Thank you.
Thank you, sir. Well, now we have reached the moment to bid adieu. And for that, Miss Hemavati from second year MBBS is here with her vote of thanks. Over to you, Miss Hemavati.
Thanks, Mr. Amok Deshmukh. Good afternoon, all. First of all, I am thankful to Almighty for giving us this glorified event. His blessings has turned this event into a grand success. I want to thank our guest speaker, Dr. V.V. Raghvendra Sai, Associate Professor, Biomedical Engineer, Department of Applied Mechanics, IIT Madras, for his active participation and giving his insight and time to us about PFAB technology. Sir, you truly made this day fruitful for us. We have no words to offer gratitude for your valuable presence in this occasion. Once again, I would like to thank Dr. V.V. Raghvendra Sai for enlightening us this knowledge. We are grateful to our Dean, Dr. Kotu sir, for providing support and valuable guidance for this program. My heartfelt thanks to Director Medical Research AVMC, Dr. Vishnu Bhatt sir, President of IIC, for his extreme support and relevance contribution in this occasion. For further thanks to our Dr. Mahalakshmi ma'am, Dean HP, for always extending her support. Thanks to Dr. Pushpa Kotur, Professor, Department of Obstetric and Gynecology, Convenza IIC, who support us more in this event. Thanks to our external members, Dr. Rekha Shetty ma'am, Managing Director, Fasta Distribution Network, Limit, Chennai. Thanks to Dr. Ali, GIEC, VMF for. Thanks to Dr. Jagadish. Thanks to Dr. SL Hoti, TCMR, Extreme Scientist at ICMR, Vector Control Research Center, Pondicherry. Thanks to Mr. Anand Sahar, MD and CEO of City Group. And also I would like to thank Mr. Manimannan, Director of Industry and Academy Relation, VMRF. I would like to thank Dr. Rajan, who always encourage us to involve ourselves in this kind of innovation programs to better ourselves every day. I would like to thank Dr. Arnachalam sir, our Vice Principal, Student Affairs, for extending his support for today's program. Also, I would like to thank Mr. Amog, Ms. Rajrani, and Mr. Karthik, our Faculty Coordinator, Dr. Aarti ma'am, Assistant Professor, Forensic Medicine, and Sibi sir, Associate Professor, Biochemistry, our moderator, Dr. Jyoti Maria Fiora, ma'am, Assistant Professor, Biophysiology, for making us this program a huge success. And finally, I thanks all the participants for being present and hope everyone got a chance to learn something new today. With this, I would like to wish you all a wonderful day ahead. Thank you all. Thank you.
Thank you, Hemavati. Now I request everyone to rise for National Anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंग विंध्य हिमाचल यमुना गंगा उज्ज्वल जलधि तरंग तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you, Dr. Sai, for once again, and we look forward to be collaborated with IIT Madras, with you, especially with your department. Thank you so much.
Thank you.
Should we close the meeting?
Yes. Yeah. Thanks.
Thanks a lot. Thank you.
Feedback. I request all participants to send their feedback. Feedback form is already there in chat box. In chat box already it is there. Please send it immediately. Okay. Thank you. Thank you, sir.
Thanks to everybody. Thank you.
Close.
Thank you. Thank you.